Hey, good morning, guys. Welcome to lecture cycle 9. We're getting close to the end of the course. There's 11 lecture cycles, so after this lecture cycle, just two more weeks of lectures as you know them. This lecture cycle is all about the topic of evolutionary relationships. So phylogenies. It's sort of a deep dive into the evolutionary relationships between different taxa. What do I mean by taxa? It is just a group of organisms. It could be, so a taxon could be a species. It could be something smaller than a species, like a single population. It could be something larger than a species, so vertebrate animals are a taxon, eukaryotes are a taxon. We're just looking at the evolutionary relationships between different combinations of living things. And using phylogenetics, once we know these evolutionary relationships, we can answer all sorts of cool questions, like who is the closest relative of whatever taxon we're interested in. So for example, who are our closest evolutionary cousins as humans. We can ask questions about how closely related we are to any other group of species. In other words, how far back in evolutionary time would we have to go to reach the most recent common ancestor of our taxa of interest? We can even answer questions such as, what did the most recent common ancestor look like? What sort of traits did it have? And so using information about phylogenetics, about evolutionary relationships, we know, for example, not only that we as humans are closer related to frogs than we are to beetles, because we have a most recent common ancestor with frogs than we do with beetles. But we can also make some informed inferences about when those most recent common ancestors would have lived. You know, how many millions of years back would we have to go to reach this guy and this guy? And also what sort of character states or traits those most recent common ancestors had. And so how do we know, why do we think, for example, that the most recent common ancestor of humans and frogs looked something like this, whereas the most recent common ancestor of humans and beetles looked something like this worm. In general, these most recent common ancestors are things that are no longer alive on the planet now. They're extinct. And for the most part, these most recent common ancestors are not preserved in the fossil record. Sometimes they are, but very often they're not. So how can we use information about character states in different groups of living things to make inferences about these most recent common ancestors, these organisms that were alive and present at these deep speciation events? And when we talk about evolutionary relationships, we very often use depictions sort of like this. We use the metaphor of phylogenetic trees. And so the species that we're interested in, or in the case of a whole life phylogeny, every single species that's alive on the planet today would be arranged. We can think of them as being like the leaves on the tree. Anything that's alive at the very tips of these phylogenies are species that are alive on the planet today. Whereas the most recent common ancestors are at these branching points of the phylogenetic trees. So using phylogeny, for example, we can ask questions about who is more closely related to whom. And so, for example, if I have like my dog and my cat, how do I know that they are much more closely related to each other than either of them is to me or to this random horse that like photobombed my field work when I was in? How do we know about that closeness of evolutionary relationships? How do we reconstruct those phylogenies, and how do we interpret a phylogeny to conclude, okay, dogs and cats are closer related to each other than either one is to, say, a human or a horse. And maybe all of these things are more closely related to each other. The humans, the horses, the dogs and cats, and the sea stars are more closely related to each other than any of them is to this octopus. How do we know? Well, in order to construct a phylogeny and use a phylogeny to make inferences about evolutionary relationships, the first thing you need to be able to do is to interpret or read a phylogeny. And so again, using this metaphor of the tree, you can see that the species of interest, the taxa of interest, here we've got a frog and a salamander, and here's Darwin representing the humans, and here's a bird. These taxa of interest are arranged at the tips of the phylogeny. You could think of them as being the leaves of the tree. And the tree also has these branching points. And hopefully you remember from last week's lecture, these branching points correspond to speciation events. So here's one branching point, and this corresponds to the most recent common ancestor of the frog and the salamander. Here's another branching point corresponding to the speciation event between the lineage that eventually gave rise to humans and the lineage that gave rise to birds. Here is another speciation event way here at the base of the tree, and we can call this one the root of the phylogeny. So the tree metaphor is strong, right? There's branching events, there's tips or leaves, and there's roots. Now, anytime you are reading a phylogeny, be aware that time flows from the root of the tree towards the tips of the tree. Okay, so these species here are species, in this case, these are all species that are alive on the planet today. And the farther back in time we go, or the farther back towards the root of the tree we go, the farther back in time we're going. So here's modern day, and we're going farther and farther and farther back until this most recent common ancestor of the whole group lived a long time ago. There's no scale on this tree, so I don't know how long ago it lived. But in general, time flows from the roots to the tips. Okay. Now, one thing that sometimes is confusing to people is that there are a number of different ways that we can draw phylogenetic trees. And so sometimes we'll have time flowing from left to right, like in this phylogeny here. Other times we might have time flowing from bottom to the top. But again, these two phylogenies are actually telling you exactly the same thing. They represent the same sequence of branching events. And in both of these, time is flowing from the root of the tree, that's a long time ago, up to the tips of the tree, which is modern day. But both of these phylogenies identify frogs and salamanders as being relatively closely related to each other, and humans and birds as being relatively closely related to each other. Okay, humans and birds, frogs and salamanders, frogs and salamanders. And so we can say that both of these phylogenies identify, say, frogs and salamanders are sister taxa to each other. They are each other's closest relative. And humans and birds on this phylogeny are also sister taxa, very close relatives. You'll notice that sometimes we can represent phylogenies using that sort of rectangular motif, like on the right. So these speciation events are shaped like that. And sometimes we have a diagonal approach to drawing a phylogeny. It doesn't matter. Both of these are telling you exactly the same thing. They're just different aesthetic choices. And yeah, this tail of the phylogeny, I could have drawn it going like this instead. So the angle of these branching events doesn't really matter. What matters is the sequence of the branching events. In other words, very far back in evolutionary time, the lineage that eventually gave rise to frogs and salamanders diverged from the lineage that eventually gave rise to humans and birds. And later, humans and birds diverged from each other, frogs and salamanders diverged from each other. So we can think of a phylogeny as being like a mobile that you'd place above the crib of a human child. So you can pivot around, you can rotate around any of these speciation events. We could flip it and have the salamander here and the frog here. Or we could flip around this node here. Or we could rotate around this node here and have humans and birds on the top and frogs and salamanders on the bottom. That's just like spinning around the nodes in a mobile. It doesn't change the information. The branching order is the same. Okay. Yes, I worked so hard on that animation. This is just showing you that the two phylogenies are showing you exactly the same information. We have the same combinations of sister taxa in both of these. Two ways of depicting the same information. And likewise, here are a number of different they're different pictorial representations of the same information. In other words, all four of the phylogenies on this slide are actually telling you exactly the same thing about evolutionary relationships. Yes, some of them are tilted on their side, so time is flowing from left to right, and some of them are facing up, so time is flowing from bottom to top. And some of them are constructed with a rectangular aesthetic, and some of them are diagonal. But they are all telling you the same thing. So E and G, for example, are sister taxa in all of these phylogenies. And C and F are also sister taxa. They're more closely related to each other than they are to anybody else. And species A is kind of the oddball. Like all the other 5 taxa are more closely related to each other. They share a most recent common ancestor with each other than any of them do with species A. Some people just look at this and they understand it immediately, and other people really need to spend some time staring at this for a while. So feel free to take the time you need with it after the lecture. Okay, but we'll do some clicker questions just to see how most of the classes do. Okay, so if I give you like these two phylogenetic trees, are they telling us the same thing? Do they convey the same information about evolutionary relationships? What do you think? Okay, well, if the clickers aren't working, we can just do show of hands because it's a fairly straightforward introduction to this. How many people think these two phylogenies are showing us the same information? Yes, okay, great job. Yeah, in both of these phylogenies, we've got birds and humans as sister taxa. And both of these phylogenies have frogs and salamanders as sister taxa. The only difference is we rotated around this node at the base of the tree. They are, oh, thanks a lot, clickers. Okay, no. Okay. Here's another example, just another set of phylogenies with 4 species or 4 taxa on them: sharks and chickens and bats and chipmunks. So you'll notice that the left to right arrangement of the taxa, both of these go shark, chicken, bat, and chipmunk. Does that mean that they're both telling us the same thing, or am I just trying to be tricky? Obviously, it's a trick, right? Okay. Nice. Okay, yeah. trying to highlight the, okay, there we go, there we go. Good job. Good job if you answered B. Yes, these two phylogenies are describing two different sequences of branching events. And the key difference here is whether the bat is more closely related to the chipmunk here, or whether the bat is more closely related to the chicken. Good. And almost nobody was fooled by the fact that I had the names of the taxa arranged in similar order. It's all about the sequence of events. Okay. One more reading phylogeny question for now. Okay, so here we've got like a number of species. There's like a shark and I don't know, a fighting fish, and this is supposed to be a salmon. Here's a frog, here's a goose, and here's a human. And so if I said, here's the frog, what's the frog's closest relative or relatives? This is a more exam level question. But I get the vibe that people will be pretty successful on it. Picking up what I'm putting down. So when we're reading phylogenies, the key thing is not the left to right arrangement of the taxa. It is the sequence of the branching events. Or how long ago was it? How far back in evolutionary time do we have to go to get to the most recent common ancestor of the frog and any of these other groups? Nice job, those who answered D. Yes, okay, perfect. D is the correct answer here. And so we know, for example, that how closely related is the frog to the salmon? Well, to get back to its most recent common ancestor with salmon, the frog would have to go this far back in evolutionary time. That is, from here to this most recent common ancestor with frog and salmon. That is how distantly related it is to salmon, whereas the frog's most recent common ancestor with the goose would be this far back in evolutionary time. It's at this point in the evolutionary tree. But this branching point here also represents the frog's most recent common ancestor with humans. Okay, so the frog is actually equally closely related to the goose and the human. We are both the frog's most recent common ancestor on this phylogeny. So we are both the frog's most close evolutionary relative on this phylogeny. Good job. Okay. Yeah, any more questions about reading phylogenies? I'll probably give out lots of practice questions as we sort of gear up for the final exam. These are the kinds of things that if you're not comfortable with thinking in phylogenetic terms right away, you can certainly work on that. And I can hand out like lots of practice phylogenies, and you can practice saying who's the closest ancestor, who are the most distantly related species on the tree. Okay, but for now, I think most people are more or less getting it. That's great. Okay, so now that we can interpret phylogenies, now that we can read phylogenetic trees and understand the information that they are conveying, we can make use of them. And you're already familiar with one possible use of phylogenetic information, and that is if you want to use the phylogenetic species concept, then recognizing which groups are each other's closest ancestors can be really, really helpful. So that is one use. But another really important way in which phylogenies are now being used is in the field of classification. So when we decide what scientific names, what scientific groupings we recognize. So If you remember the Life on Earth lecture, and I was saying I don't like the term monkeys. Monkeys is a stupid term. I also don't like the term fish, and reptiles also does not make evolutionary sense. The reason that I had it out for all of these groups, all of these terms that we use, was because some terms, some classifications that are widely used do not reflect evolutionary relationships. And most evolutionary biologists believe that group names, the names that we give a group of taxa, should actually reflect those evolutionary relationships. In other words, if we're going to name a group, say the monkeys, then everything that we include as part of the group needs to be more closely related to everything else that's called a monkey than it is to anything that's not called a monkey. So group names should reflect evolutionary relationships. And if you've done the pre-reading, you're already familiar with the terms monophyletic groupings and non-monophyletic groupings. But there's a school of thought called cladistic analysis, and the cladistic method of classification argues that, thanks a lot, clicker, argues that only monophyletic groups should get names. This is another way of saying every member of a group should be more closely related to every other member of the group than it is to anything outside of the group. It's just a shorthand, a more efficient way of saying the same information. And so what's a monophyletic grouping? Well, first of all, if you've done the pre-reading in the textbook and you've seen these little diagrams, you may remember that the textbook actually distinguishes between monophyletic groupings and paraphyletic groupings and polyphyletic groupings. I don't actually care if you remember the difference between paraphyletic and polyphyletic. I don't care at all. All I want you to recognize is whether or not a group is monophyletic. And a monophyletic group is a group that includes all of the descendants of the group's most recent common ancestor. So if I wanted to, okay, so here we've got here's evolutionary relationships here, and all of these species on the top row are modern day species that are alive today. If I wanted to recognize a group corresponding to this blue circle, so I want to say, okay, the blue group, I'm gonna assign that a name, assign that a taxonomic name. That would be fine because the blue group is monophyletic. So we've got our two species of interest that we want to include in the group, here and here. And we say, okay, what's the most recent common ancestor of those two species? Okay, it's this guy here. Does the grouping include all of the descendants of that most recent common ancestor? And in the case of the blue circle, yes, it does. Now, just as a quick little digression, sometimes people will get bent out of shape as to whether a monophyletic grouping has to include that most recent common ancestor or whether it can just include the modern day descendants. I don't really care. The important thing, from my point of view, is that a monophyletic grouping is something that includes all of the descendants of the most recent common ancestor. So grouping these two blue species together would be fine. That's monophyletic, and they are more closely related to each other than they are to anything not in the blue circle, so that's monophyletic. We could also call that a clade. A clade is just another word for a monophyletic grouping. A group in which everything inside the clade is more closely related to anything that's not in the clade. Okay, contrast this. What if we wanted to group these two species together and call it the purple group? That would not be a monophyletic grouping. And it's not that these two species don't have a most recent common ancestor, but their most recent common ancestor is all the way down here at the base of the tree. And so then if we say, okay, does the proposed group include all the descendants of the most recent common ancestor? Well, the answer is no. So here's the most recent common ancestor, and this descendant and this descendant are left out of the purple group. And so this purple grouping is not monophyletic. It's not a clade. And so trying to assign a name to that group without also including these two species would not reflect evolutionary relationships. So that would be problematic. And likewise, if we wanted to include these three taxa, but not this guy, and call it, say, the orange group, that would also not be a monophyletic grouping. And again, the problem with this is that, yeah, we can go back far enough in time and find the most recent common ancestor of this guy and this guy and this guy. It's all the way down here. But you can see that not all of this guy's descendants are included in the orange group. So this is not a monophyletic grouping. This is not a clade. Only the blue circle counts as a clade. Okay. Oh yeah, this is just an alternative little mnemonic device or thought experiment that you can do. If you're trying to determine whether or not a proposed grouping of organisms, a proposed grouping of taxa is a clade or is monophyletic, then you can use this thought experiment. Okay, this is for people that really love the tree metaphor, that like the tree of life, and they want to just lean into that and pretend that these are like literally branches of a tree, and you could cut branches off. If you can isolate a grouping with a single clip of your scissors like that, then it's a clade, then it's a monophyletic group. Okay, so if we've got crocodiles and birds and we want to group them together, we could just cut anywhere along this lineage here, make a little snip, there we go, and we've got a little bouquet with the crocodiles and birds together. So that tells us that crocodiles and birds are a monophyletic grouping. Whereas if you need to make more than one snips of the scissors to isolate the group that you want to call a group, then it's not monophyletic. And so if we wanted to put lizards and crocodiles together and call them, say, the reptiles, as indeed traditional classification has done, we would have to snip once here, but we'd also have to snip again here to get rid of the birds, because birds aren't considered reptiles. And so this is how we know that lizards and crocodiles, so-called reptiles, do not actually form a monophyletic grouping. There are some things in that group that are actually more closely related to birds than they are to other reptiles. Questions on that so far? Yes?
So it's always just to the most recent common ancestor?
Yes.
Or did you go on, well, not the like my lab, if you wouldn't go
That's
down
it?
to where the lizard or mammal is just the most common.
Right, yeah. So if we just want to group crocodiles and birds together, then we just have to go down to their most recent common ancestor, to the crocodile bird most recent common ancestor, yeah. And say, okay, are all the descendants of this guy represented in the grouping? And yes, they are, yeah. Whereas if we wanted to group lizards and crocodiles together, we'd have to go down, here's the lizard crocodile most recent common ancestor, and all of the descendants of this guy are not represented in.
So that if you did like lizard and bird,
Lizards and birds, but leaving out crocodiles? Lizards and birds, leaving out crocodiles would also be not monophyletic, because you'd have to do, that's like an extra snip, yeah. Yes? Yes, yeah, a single speciation event, yeah.
Like technically lizards, crocodiles, and birds would be part of a single monophyletic.
Absolutely, yeah. You could totally recognize lizards, crocodiles, and birds as a clade. They are all more closely related to each other than any of them are to mammals or anything else, yeah. Great, okay, awesome. Oh yeah, okay, another practice question from a previous year's final exam. So yeah, just to practice your ability to recognize monophyletic or non-monophyletic groups, you would get a question sort of like this. According to this phylogeny, which combinations of taxa, which combinations of letters are monophyletic? I'll give the clickers like 10 more seconds to show up. There they go. I think the Wi-Fi in this room is like the worst that it is anywhere on campus. Nowhere else is it as bad as in this room. Raise your hand if you need another like 15 seconds or so. Okay, we're good to take it up? All right, let's see. Oh, you guys, yeah, awesome, awesome, awesome. Yes, A is the right answer, so very nicely done. Let's match that. Okay, yeah. U and V are monophyletic. We could make just a snip here and isolate U and V, and that's fine. T, U, and V are monophyletic. They're all more closely related to each other than they are to anything outside that group. The whole thing, R, S, T, U, and V, is monophyletic. We could like make a snip here. This is a clade. The only non-monophyletic grouping is if we try and include S, T, U, and V, but exclude R. That would not be monophyletic because the most recent common ancestor of S, T, U, and V has one descendant who is not represented in the group. So well done. Okay. So humans have been really interested in naming things and classifying the worlds around us, and actually scientists started doing that hundreds of years ago, long before we knew about evolutionary relationships and had worked out these cladistic principles. And so for that reason, there's lots of classifications. There's lots of scientific names for things that actually don't represent clades, that don't actually reflect evolutionary relationships. And so things like the prokaryotes, for example, the bacteria and the archaea, you can see that this is not a monophyletic group, even though eukaryotes do, in fact, happen to be monophyletic. Prokaryotes are not monophyletic. If you've heard of the distinction in plants, we talk about monocots and dicots. Monocots are a monophyletic group, but dicots are not. There are some things that we call dicots that are actually more closely related to monocots than they are to other dicots. And here, fish. You've already heard what I think about fish. There are some things like a coelacanth or a lobe-finned fish is more closely related to an amphibian or a mammal or any other tetrapod than it is to other things that we also call fish. So lots of groups that are still recognized but are not actually monophyletic. So yeah, prokaryotes, reptiles, monkeys, if we don't distinguish Old World and New World. I don't know if they still teach the 5 kingdoms in like public school anymore. Do they still teach the 5 kingdoms? Yes, okay, so not all of those kingdoms actually reflect evolutionary relationships. Kingdom Protista, that's not a clade. Kingdom Monera, that's not a clade. So, yes, just another more slightly complex example. Terms like foxes or wolves or wild dogs or jackals, all of these turn out not to be monophyletic either. In other words, you don't have to memorize this phylogeny, but just know that there are some things that we call foxes that are actually more closely related to things that we call wolves than they are to other things we call foxes. So lots of groups have names, but those names don't reflect evolutionary relationships. This is changing, but there's a lot of inertia as well. So phylogenies can be useful in taxonomy and in recognizing which groups should be named and considered members of the same group. Okay, so now that we know the sort of ways in which we can use phylogenies, people are probably starting to wonder, how do we know what these evolutionary relationships are? That is, how do we build a phylogeny? Or if there's 2 or more possible phylogenies, possible sets of evolutionary relationships for a group of taxa. How do we know which one is right or which one is probably right? So building and evaluating phylogenies. It's down, it's up. That's fine. Okay, so basically, in a perfect world anyway, if everything were really, really great from the point of view of somebody who's interested in classifying life and working out evolutionary relationships, we could just always use similarities, okay? So the more similar two taxa are, probably the closer related they are. And so we could say, for example, okay, here's like this bird and this chameleon and this sea turtle, and maybe I want to work out evolutionary relationships among these three things, which one is most distantly related to the other two. I could say, well, let's see, birds have hollow bones, but the middle guy and the right guy have bones that are solid and heavy. So the bird's a little bit different. The other two have a trait in common. I could say birds have feathers, but these guys both have scales on their body. I could say birds are endotherms, so they're warm-blooded, and these two are cold-blooded. There seem to be a lot of similarities that, say, the chameleon and the sea turtle have. But in fact, birds and chameleons are more closely related to each other than either of them is to the sea turtle. So actually, these two are closer related to each other than either one is to these guys. And this raises an important point, and that is that not all similarities are equally informative. Not all similarities give us useful information about evolutionary relationships. And so, yeah, sort of a similar assemblage of species, but now we've got turtles and, what's that, a gecko, and crocodiles and birds. And again, there's many similarities that these three have with each other. So the fact that they are all cold-blooded, the fact that they all have scales, the fact that they have solid bones instead of hollow bones. For these reasons, early people trying to do classification sort of grouped all the reptiles together and said reptiles belong together and the birds are more distantly related. But in fact, using more modern methods, it turns out that, yeah, birds are actually more closely related to crocodiles than these guys are to crocodiles. And all of these guys are more closely related to each other than any of them are to the tortoise. So some similarities turn out not to be that informative. Some similarities might actually mislead us and make us jump to wrong conclusions about evolutionary relationships. Yeah, likewise, if you were trying to work out evolutionary relationships among this group of species, you might say, all right, this guy has bilateral symmetry, and these two taxa have radial symmetry. These two taxa don't have a backbone, but this taxa does have a backbone. These two taxa live in the water, and this taxon lives in the air. But again, patterns of relatedness actually put the sea star and human as being sister taxa. They're more closely related to each other than either one of them is to the jellyfish. So not all of these traits are actually useful. Not all traits provide useful information about evolutionary relationships. And so this is where the field of cladistics comes in clutch, because it sort of points out the type of similarities that we should be paying attention to when we are trying to reconstruct evolutionary relationships. So imagine that we are trying to work out evolutionary relationships between these 6 taxa of vertebrates. Imagine you don't already know the evolutionary relationships, but you're trying to work them out. You can imagine, for example, that there could be a trait that is just found in a single taxon. Okay, so yeah, birds have feathers, and none of the other group have feathers. Is a trait like this, with this distribution, is that going to be helpful to you in working out the evolutionary relationships between these groups? Birds have feathers. Does that help me recognize which other taxa are more closely related to birds? No, it doesn't. Birds have feathers, but that doesn't help me at all. And we call this type of trait an autapomorphy. An autapomorphy is not useful in reconstructing evolutionary relationships because it's not really a similarity, right? It's not a similarity that birds share with anything else. It's just, yeah, birds have this, and nobody else does. It is unique. Having feathers is unique. Only one of the taxa of interest has that trait, and so we can't use it to group taxa together. Yes?
Would you think that it aligns mostly with the morphological species concept as opposed to the other species concept?
Okay, yeah, so does this mean that I'm using the morphological species concept? That's a great point. We can use traits that are morphological, like feathers or solid bones versus hollow bones, or we could use genetic traits like DNA. Do they have a C or a G at this point in their genome, or do they have this insertion, or don't they? We could use genetic information, or we could use morphological information. The principles are the same, and actually people use both types of evidence. Yeah, good question. Okay, so birds having feathers is not informative because it is not shared, it is unique. An autapomorphy like this is also, seems to be of relatively recent evolutionary origin. In other words, we call having feathers, that's probably a derived trait. In other words, probably the most recent common ancestor of this whole grouping did not have feathers. This is probably something that just evolved in the lineage leading to the birds. More on traits how to recognize traits that are derived versus older evolutionary traits coming up. So autapomorphies, like that are found in one taxon but none of the others, those are not useful to us in reconstructing evolutionary relationships. Okay, so imagine then that we say, okay, well, I know that all of these things are made out of cells, or even I know that all of these things have a backbone, have vertebrae. Is a trait like that's present in all the species we're looking at, is that going to be helpful to us in classifying and working out their evolutionary relationships? No, because it's too broad. Traits like these are what we call simplesiomorphies. Simplesiomorphies are traits that are, well, they're shared, so they don't have the problem of the autapomorphy, but they're shared by two or more of the groups of interest, but they are also ancestral, or they're probably ancestral. So most likely, the reason why all 6 of these things are made out of cells is probably because their most recent common ancestor was also made out of cells. So traits like this that are of very old evolutionary design, that is, are ancestral, that is, they were probably present in the most recent common ancestor of the group. Even though they're shared, we don't consider them informative. It doesn't really help us. But a trait like this, imagine that we say, okay, birds have like a hard shell to their eggs, and so do crocodiles, and nobody else does in this group. That is actually the type of trait that is informative. This is the kind of similarity that we are looking for. And we call this a synapomorphy. So a synapomorphy is a trait that is shared. That is, it is present in more than one species that we're looking at. So crocodiles have it and birds also have it. It's shared and it's probably derived. That is, we're going to infer that probably the most recent common ancestor of crocodiles and birds had that trait, but the most recent common ancestor of the whole group probably did not. So we think that this trait, the having hard shells to your eggs is probably derived. That is of relatively recent evolutionary origin. Okay. And so, according to cladistic principles of classification, the only similarities, the only traits we should be paying attention to are traits that are synapomorphies, traits that are shared and derived. And so using synapomorphies, we can reconstruct evolutionary relationships between taxa. So yeah, so here's feathers. Here's a trait that is not a synapomorphy across these 6 or 7 taxa. It's just only found in the birds. But here, having an eggshell with a hard shell, this is a synapomorphy that unites crocodiles and birds. And there is actually a term for the crocodile bird clade. They're called the archosaurs. So this is a synapomorphy that unites these two. This tells us that crocodiles and birds are more closely related to each other than they are to anyone outside that group. And having like fingernails or claws, like nails on your digits, this is a similarity that unites, say, mammals and lizards and turtles and crocodiles and birds, but it's not shared with, say, frogs and salamanders. So that's another synapomorphy. So synapomorphies are things that are shared and derived. And so, yeah, in cladistics, only synapomorphies are informative. We should only pay attention to traits if they are shared and derived. Now, what you're probably thinking is it is easy to tell whether a trait is shared or unique. Like you just have to count, and if you count past, if more than one taxon has it, then it's shared. So that's fine. It's more challenging, maybe, to infer whether a trait is derived versus ancestral. In other words, before we know the evolutionary relationships between, say, those different taxa of vertebrates, how do we know what the most recent common ancestor of the group had? So how do we figure out whether, having fingernails is probably a thing of recent evolutionary origin, or how do we know that having fingernails wasn't present in the most recent common ancestor of the whole group, right? You're probably wondering. And the way that we answer that, it may be a little unsatisfying because we can't actually answer it with 100% certainty very often. But we can make a very informed inference. And that is if we use a technique known as outgroup analysis. So an outgroup is defined as something that is a close relative of the group that you're trying to classify, but however, we know it is not a part of the group that you're trying to classify. And so, for example, if we wanted to work out evolutionary relationships between species A, B, C, and D, these are our taxa of interest. These are members of the in-group that were interested in their evolutionary relationships. It's often very useful to have another species that is a fairly close relative, but we're sure it doesn't belong to the in-group. And so we'll pick up at this point in Wednesday's lecture. Thanks.